What are we learning today?
We are going to learn about alcoholic fatty liver disease.
I know that we have learned that alcohol is bad for us, but how does it cause alcoholic fatty liver disease?
Overconsumption of alcohol results in the buildup of triglycerides and other fats in liver cells, resulting in alcoholic fatty liver disease, or ALD.
How exactly does ALD impact the liver?
In a healthy liver, alcohol dehydrogenase plays an essential role in the metabolism of alcohol by reducing NAD to NADH. When alcohol dehydrogenase is impacted by excess alcohol consumption, the NAD-NADH ratio is altered and promotes fatty liver by inhibiting gluconeogenesis and fatty acid oxidation. This is what leads to excess fat in the liver.
Which proteins are affected by alcohol and alcoholic fatty liver disease?
A group of proteins that is affected by alcohol and influences fatty liver disease is sterile regulatory element binding proteins, or SREBPs. SREBPs are membrane-bound transcription factors that maintain lipid homeostasis. SREBPs activate the expression of more than 30 genes and affect the synthesis and uptake of cholesterol, fatty acids, triglycerides, phospholipids, and the NADPH cofactor.
What are SREBPs?
SREBPs are part of the helix-loop-helix-leucine zipper family of transcription factors. They differ from other proteins because they are synthesized as inactive precursors bound to the endoplasmic reticulum. Each inactive form of SREBP is made of 1,150 amino acids and organized into three domains. The first is an N-terminal domain of 480 amino acids that contain the helix-loop-helix-leucine zipper region for binding DNA. The second is two hydrophobic transmembrane spanning segments interrupted by a short loop of about 30 amino acids in the lumen of the ER. The last is a C-terminal domain of 590 amino acids that performs the regulatory function.
What do SREBPs do?
Specific to the liver, 3 SREBPs regulate the production of lipids for export into the plasma as lipoproteins and into the bile as micelles.
What is the mechanism of SREBPs?
In order to reach the nucleus and act as a transcription factor, the NH2 terminal domain of each SREBP must be released from the membrane proteolytically. 3 different proteins are required for SREBP processing. One is an escort protein designated SREBP cleavage activating protein. The other 2 are proteases, site 1 protease and site 2 protease. Newly synthesized SREBP is inserted into the membranes of the ER, where its C-terminal regulatory domain binds to the C-terminal domain of the SREBP cleavage-activating protein. SREBP cleavage-activating protein escorts the SREBP from the ER to the Golgi apparatus, where it is processed by the proteases. In the Golgi apparatus, site 1 protease cleaves the SREBP, dividing the SREBP molecule in half. The N-terminal helix-loop-helix-leucine zipper domain is then released from the membrane via a second cleavage mediated by site 2 protease. The N-terminal domain, designated nuclear SREBP, translocates to the nucleus where it activates transcription by binding to the promoter or enhancer regions of multiple target genes. The enzymes involved in fatty acid synthesis are controlled by sterile regulatory element binding protein 1, or SREBP1. Ethanol exposure significantly increases the SREBP regulated transcription by increasing the amount of mature SREBP1 proteins. Thus, ethanol increases the fatty acid synthesis through SREBP1. The effect of ethanol on SREBP1 is likely mediated through its metabolism to acetaldehyde. Too much fatty acid synthesis in the liver eventually leads to the pathology of fatty liver disease.
Can we target an SREBP to treat ALD?
There are several inhibitory mechanisms being studied for the treatment of ALD. One such inhibitor, botulin, has a suppressive effect on SREBP maturation and development. This suppression results in a decrease in cholesterol and fatty acid synthesis. Another direction of treatment by inhibition also seems to be promising. The SREBP contains many different parts, as we discussed before. 2 genes within SREBP are SCAP and S1P that are liver-specific genes. A study in mice found that disrupting the functions of these genes dramatically reduced the N-SREBP1 and N-SREBP2 levels in the liver. This diminished the expression of all SREBP genes in the cholesterol and fatty acid synthetic pathways. The synthesis of both cholesterol and fatty acids fell by 70 to 80%. This model gave in vivo evidence that SCAP activity in particular, when inhibited, reduced the production of fats in the liver that normally build up as a result of ALD. A great quantity of research is currently being performed on ways to reduce the fatty acid and cholesterol buildup in the liver as a result of ALD. Although we are currently making leaps of discoveries in treatment of ALD, we still have a long way to go. This lesson may be motivating one of you to continue the research and finally find a treatment to ALD.